Okay, lecture 8 is the kind of continuation of the lecture 7. So we're going to talk about the PCR continuously. So through the lecture 7, we learned about the PCR principles, how it works, and what is the science behind of it. And now through the lecture 8, we're going to understand where you're going to use the PCR principle. Okay, and what are the main application of PCR technology? Okay, so number one application is, of course, you can make more DNA molecules. So enzymatic amplification of DNA. So basically you are making a bunch of copies of DNA from a very small amount of template DNA you have. You will make billions of copies. So that is the number one application of PCR, of course. And then number two will be the direct DNA sequencing of PCR. So DNA sequence we'll talk about in the next lecture. But in the old sequencing method that's developed by Sanger uses dideoxynucleotide. In that method, there's a DNA replication involved. So to carry out the DNA replication inside of a test tube, yes, the PCR process is used. So yes, DNA sequencing will use the PCR amplification process. So you'll have to add a primer. In this case, a single primer around the region of your DNA sequencing area is added, and then the nucleotides that are labeled with fluorophore will be added there. So you can identify each nucleotide sequence from your reaction. So that's how it works. So next lecture we'll talk about the DNA sequencing reaction, and you will understand how it's done. Okay. So yeah, genomic sequencing process is illustrated in this diagram. So starting from 1990s, there are so many genomes that have been sequenced out. And it's done through this method. Genomic DNA was isolated, and then there are PCR-based sequencing method that has identified all the sequence of the genome, and then assembly of those sequences are done using a supercomputer. That's the process of DNA sequencing. And then molecular cloning of PCR products. So like I told you in the previous lecture, Taq polymers tend to add one extra nucleotide at the end of its PCR cycle. That nucleotide is AN, right? So there's an A overhang in the PCR product. So if that is the case, what do you do with that A overhang? If you have a plasmid DNA with a T overhang, you can nicely insert your PCR product in that. We call that a T and A matching cloning process. That we call the TA cloning process. So you can do that. You can clone your PCR product into a plasmid DNA using that feature, or you can also, that process is probably illustrated in the TA cloning. This is the method. So using that A overhang in the PCR product and then insert that into the plasmid DNA with the T overhang. That's a TA cloning, right? And another way of cloning your PCR product is, if you can maybe cut out that extra A from your PCR product, or you can maybe fill that extra A with a T nucleotide. So to make that PCR product as a blunt end DNA fragment, then you can do a blunt end ligation, right? So you can either cleave it, cut it up, cut A off, or you can fill that A with a T to make a blunt end. Okay, once that's successful, so you use that enzyme to do that, and then you can ligate the blunt end PCR fragment into the plasmid DNA with the blunt end as well. It's a blunt end to blunt end ligation. You can do that. Okay, so this diagram shows you how you can do that. So that's the PCR product with A overhang, right? So and will be repaired. So you can maybe add a polymerase enzyme to add extra T there. So now it's TA match. So there's the blunt end there. So there's the plasmid DNA is also cut with the enzyme that creates a blunt end. So it's blunt end cutting enzyme, and then you can have the blunt end to blunt end ligation. Once that's recombined, then you can put that into a E. coli cell. This transformation will take place, and you can look for the cells that have the insert of this recombinant DNA. So that's how you can do the cloning. Okay. Another cloning method will be generating a cohesive end at the end of your PCR product. Okay. How do you do that? In this case, you will use the primer sequence to add extra enzymatic cutting site in your PCR product. So what I mean is, so here is your primer, maybe first primer binding on one of the two strands. There's another primer that binds on the other strand, right? On the primer sequence, yes, let's say about 20 nucleotide that binds on your template DNA. Same goes here. But on top of the 20 nucleotide, you maybe add about 6 nucleotide base pair extra, but that's extra sequence that doesn't bind on the target DNA. So it's going to overhang, it's going to stick out because it's not binding, it's not making the complementary base pair. Same way with this. But since it has 20 nucleotide long sequence that is annealing, so that is enough for it to recognize the target DNA template strand and start annealing. And then once they anneal, then Taq polymerase grabs the end of the tail and start the replication. So yeah, PCR will work without any problem. And if we repeat this a few times, yes, you can generate PCR product that has the primer sequences embedded in them, right? Then the question is, why did you have to add that extra sequence there? The reason is because the extra sequence include enzymatic cutting site. Let's say Hindi3 cutting sequence is added there, maybe PST1 or PBO2 enzyme cutting sequence is added there. Now this PCR product has those enzymatic cutting sequences. Now you can treat this PCR product with those enzymes. Then once the enzymes cut, then now you have cohesive bands generated. So now you have a cohesive band here with Hindi3 sequence. This you have cohesive band here with the PBO2 sequence sticking out, right? So PCR with 2 enzyme cutting sequences, they are cohesive bands. Now if you can cut your plasmid DNA with those 2 enzymes as well, Hindi3 and PBO2, now they have matching cohesive bands. So they will be matching up nicely. So this fragment will be inserted right there through the perfect match. You ligate it, and now you have that recombinant DNA made. And then recombinant DNA can be transformed into E. coli cells, and you can screen for the right cells that have taken up the recombinant DNA. So same process will happen here. Okay, so that is called the cohesive end ligation. By adding extra sequence on your primer sequence, that will be recognized by the enzymes. So that extra sequence is used for cloning purpose. So there are 3 ways of cloning your PCR product. Okay, so number 1 is blunt end ligation. Number 2 is cohesive ligation using the extra enzyme cutting sequences. Number 3 is the simplest, TA cloning kit, because your PCR product comes with a extra overhang. So just insert that into the T overhang plasmid DNA. So that's going to be the cloning process. 3 different methods, right? And then there's another method. Oh, this is another application where the PCR is used. That is enzymatic application of PCR in the process called RT-PCR. RT-PCR means what? So reverse transcription PCR is what RT-PCR means. So you can amplify DNA out of RNA template. That's what reverse transcription is. Okay, why do you need to do that? When you try to screen if some genes in your genome are being expressed or not. Okay, so when I say if a gene is expressed or not, gene expression means what? Your gene sequence is used to make maybe RNA, and then maybe the RNA may be used to make protein. So that central dogma processes, transcription, translation, is that happening inside of a cell or not on the particular genes, right? So that's how you can, use this PCR, because once the transcription happens, you will be able to make bunch of RNA molecules. Okay, from those RNA, you can make a DNA copy. That's what reverse transcription is. There is a special enzyme that can do that. We call that reverse transcriptase, right? So by doing so, if we can make bunch of cDNA, that means complementary DNA from RNA molecules, that means yes, that gene is being expressed. So that's the screening of gene expression pattern. Okay, so the process is, first, you will treat the cells with the condition where you think the genes will be expressed. Okay, so certain condition will trigger the gene expression. Okay, if you want to see if that hypothesis is true or not, okay, then after exposing cells to that condition, then you're gonna isolate RNA molecules from those cells that are treated with the condition. Okay, so you'll have to collect some cells right away and break open the cells, isolate cellular RNA molecules. Okay, and then from those RNA molecules, you may have to fish out mRNA molecule. There's a certain way of doing that. Okay, and if you don't have to fish out, that's fine. If it's a eukaryotic cell, you don't have to do that. Then you can add the primers that can bind on those RNA molecules. Okay, so these primer, again, a single strand DNA molecules, they can have a matching sequence to RNA molecules, and they will anneal. Okay, once they anneal, and then you do the kind of cooling. Okay, RNA molecules binding with the primer. Okay, you can actually heat it and cool it down. Okay, so then that hybrid RNA with the primer is exposed to an enzyme called reverse transcriptase. Reverse transcriptase enzyme can make DNA out of RNA template, right? So if you can keep the temperature at 42 degrees 1 hour, that will be enough duration where enough RNA transcription so reverse transcription can take place. So you will make bunch of DNA from RNA. So we call those DNA cDNA, complementary DNA. They have the complementary sequence to RNA molecules. That's the reverse transcription that take place. Once that's done, and then you can add another PCR primer that will bind on the cDNA, and you can do the amplification again. So now you can make double strand DNA molecules from cDNA. Okay, so that's the end of the process. Okay, if you don't understand that, maybe you can go to this website, just copy and paste that website on your browser. So that was the website. Okay, if you come here, you can play this nice animation. So if you play that, to perform RT-PCR, you need all these reagents. Okay, so what is that? The buffer you need, of course, and nucleotides, and then you need DNA primers, and then you need enzymes. Okay, Taq polymerase enzyme and reverse transcriptase enzyme and nucleotides. Okay, and then RNA molecules that you isolate from the cells are added. Okay, incubated 37 degrees. So let us see what happens inside of a tube. Okay, so RNA molecule, a whole bunch of them, right? And there's a primer that binds on the RNA. Okay, and the reverse transcriptase enzyme and DNA nucleotides. Let's see what happens. So that's the primer binding on the RNA molecule, and then the reverse transcriptase binding on that. Okay, that's a reverse transcription happening. So there you go. That's a cDNA just made from RNA, right? Now you have the cDNA just made. Now you have a second set of a primer that can bind on the cDNA. Okay, let's see. There you go. Now that's a regular Taq DNA polymerase binding on the tail of the primer. Then there's a regular DNA replication. Now you made a double strand cDNA molecule. That's the reverse transcription, and followed by the replication. So you repeat that so many times, you have a bunch of copies made. Okay, so PCR continues over. So you can make billions of copies. You can analyze the result. Okay, so that's that. Okay.